Hello students, hope you are safe. In the previous session, we learnt about heredity and variation and Mendel's contribution to the field of genetics. In this session, before learning about Mendel's experiment, let us get acquainted with genetic terms and symbols. Why do we study the genetic terminologies? It is important to understand the terms used in the experiments in order to understand the experiments better. This will further help you to understand the principles that were postulated on the basis of Mendel's experiment. Let's begin with our genetic terminologies. The first term, character. Here, it refers to a general feature of an organism such as eye color, plant height, etc. These are those features which you can observe, which are visible to our eye. Dominant character. The characters which express itself in all the members of the first filial generation from a cross between two pure breeding parents, differing in respect of this character. It is called as dominant character. Dominant character is seen when an allele expresses its trait even in the presence of an alternative form of an allele. Let's have a look at the recessive character. The suppressed character which does not appear in the first filial generation is called as recessive character. Have a look at this picture here. We have two dominant parents, that is, here we are considering the color of the flower. A purple male flower and a purple female flower. So the allele that is going to be expressed is going to be purple. But when you take one of the characters, that is a purple flower, and the other as the white flower, yet the offspring is going to appear as purple. This shows that the white character is a recessive character. Then when can you see the expression of a recessive trait? If both the parents have the recessive character, that is both the male and the female plant has white flowers, only then will the offspring exhibit the recessive character and show the white flowers. Trait. An inherited character and its detectable variant is called as a trait. Example, tall, dwarf. Look at this picture again. We have purple flower, we have white flower. These are called as traits. So a character, that is the flower color, that is purple, and you have a detectable variant, a different allele, which is a white flower. Factors. It is a particular segment of DNA which is responsible for inheritance and expression of a character. It's a unit of heredity, a particle present in organism which is responsible for inheritance and expression of a characters. Factor is the one that is passed from one generation to the next through gametes and not a character itself. Have a look at this picture here. In your free time, you can consider looking at the different traits that you have and the traits that you have inherited from your parents. For example, the color of your eye, the color of your hair, your height. These are some of the features which are called as characters you observe in yourself, and you can compare with your parents for showing the presence of these characters in them. The next important term is gene. It is a particular segment of DNA which is responsible for inheritance and expression of a particular character. The term was first coined by Johansen. Genes is the shortest segment of DNA responsible for the expression of its specific character. Gene is also called as the basic unit of heredity. It is basically a small DNA segment that determines a biological character of an organism. Gene is situated on a chromosome. Have a look at this picture here. You have your chromosomes. If you open the chromosome up, you will get your DNA segments, and you will find a particular region which has information for a particular character. For example, your height. The red mark gene is that region of your DNA which will decide and determine the height that you are going to have. This region is called as a gene. The next term is genome. It is the total genetic composition of an organism. So you have your entire set of chromosomes. This whole set is called as a genome. This has a complete information about all the genetic characters that you have, both the one you have expressed and the ones which are suppressed. Moving to the next important term that you need to know is alleles. An allele is one of a particular form of a gene. Allele is short form of allelomorph. The term was coined by Betson and Saunders in 1902 for characters which are contrasting or differing from one another. Allele is an alternative form of the same gene in which one is parental and other is maternal. In case of plant height, for example, tallness and dwarfness are two alleles of a gene. A pair of alternative corresponding genes that are occupying the same position or a locus of the homologous chromosomes are called as alleles or allelomorphs. Alleles basically are two alternative forms of a gene or a factor, and they are called as alleles of each other. They occupy identical loci. Loci or locus means positions on homologous chromosomes. These terms were first coined by Betson. Have a look at the diagram here. You have a gene which says it's a gene for color of the eye. And what are the alternative alleles? Brown, blue, emerald, gray. This is basically, we are talking about the human alleles. We have many alternative forms for a particular genes. When it came to Mendel's just had two contrasting characters. For example, for his height, he had tallness and dwarfness. Here we have another example of a hair color, gene for hair color. You have different types or alleles which can determine the color of your hair. You have blonde, red, brown, black. Coming to the term locus, the position on a chromosome where an allele is located is called as a locus. Here, look at this figure here. This is your chromosome and these positions are called as loci. At this region of a particular chromosome, you have the genes located for the color of your eye. Here you have the gene located for the color of hair. For example, this chromosome has a gene which when expressed for your eye color will give you brown color trait, and the hair will give you, again, you will have slight brown or blonde color hair. In order to give you a clear idea about allele and locus and genes and alleles, I have put a distinguish in front of you where the concepts of allele, locus and genes might become even more clear. Discussing the first important difference, an allele is an alternative form of a gene that is arisen by mutation and is found in the same locus of the homologous chromosomes. A locus refers to a position of an alternative allele on a chromosomes. Allele is a nucleotide sequence, whereas locus serves as markers. When you talk about genes and alleles, gene is a segment of DNA which controls a specific trait, and allele is a variant of that particular gene. A single gene determines a particular trait. Two or multiple alleles bring variation to a particular trait. You can pause the video here and go through the points that are jotted to have a clear idea about these terms. Quickly moving to the next important terms, that is phenotype and genotype. Phenotype is the physical and observable expression of a character, and genotype is the genetic expression of a character. These terms were first coined by Johansen in 1909 for the visible characters or the external appearance of an organism with respect to a particular character or a group of characters. It is the observable characteristics in an individual. Though the phenotype is determined by the genotype, but in certain genes do not express for a number of reasons. So the genotype and phenotype of an organism may not be the same. When you talk about the genotype, it's the genetic constituent of an organism. For the plant height, capital T, two capital T's, capital T small t, and two small t's are the different genotypes. The genotypic ratio of F2 in monohybrid cross is 1:1:1. It is the observable phenotype. Here, have a look at this picture. You can see the different characters, and this picture here gives you an idea about the phenotype and the genotype of a particular plant. Here we are taking the example of the flower color, purple and white. This is the phenotypic expression, and over here you have the genotypic expressions, which will give you an idea about the different types of genotype that are available. Homologous chromosomes and heterozygous. Now you have homozygous condition and an heterozygous condition. A diploid individual carrying two identical alleles is known as homozygous. It is pure for that trait or character. Example, capital T's, two T's are the identical alleles for tallness. Similarly, small two T's are the identical allele for dwarfness. Basically, you denote the dominant characters with capital letters and the recessive characters with the small letters. Heterozygous condition, a diploid individual carrying two different alleles are known as heterozygous or hybrids. It is impure for a trait. Homozygous are pure for that particular trait. So you have one capital T, one small t. Here in this picture, you can understand two capital B's. When both the alleles are same, they are called as homozygous. When both the alleles are different, they are called as heterozygous. Hemizygous, when a genotype is determined by a single gene or a factor. Pure lines, it is a variety with a homozygous, which is homozygous in condition. It is called as a true breeding varieties. That is, it has either both the alleles that are dominant or both the alleles that are recessive. Hybrids are the products of a cross between two genetically different individuals belonging to the same genus or species or between two different genera. These are called as hybrids. Basically, when you talk about hybrids, they are products of a cross between two dissimilar parents having contrasting traits. Hybridizations, it's a phenomenon where you speak about mixing of two unlikely genetic constituents. In Mendel's experiment, you will come across the term monohybrid cross, dihybrid cross, polyhybrid cross. Monohybrid cross, it's a cross between two parents that are differing in just one pair of alternating or contrasting characters. In this type, single character is used. For example, in his cross, where you talk about the height, a tall plant was crossed with a dwarf plant. This is called as a monohybrid cross. The dihybrid cross, a cross between the two parents that are differing in two pairs of alternative characters, is called as a dihybrid cross. In this type, two contrasting characters are used. For example, seed shape and seed color. So you have yellow round versus green wrinkled. Polyhybrids, it's a cross between two parents differing in more than two sets of alternative characters. There you go. Thank you, students, for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe, and keep on learning. Thank you.